symbiotic cell is walking on the street and an alien attacks it. So now it needs a new cell to take its place that isn't damaged. Before the cell can begin to replicate, the DNA must first be replicated. DNA is made up of nucleotides that are made up of 5 carbon sugar molecules, a phosphate group, and 4 different nitrogenous bases. DNA must be split first to make 2 new strands. Helicase unzips DNA strands and creates a replication fork. To keep the newly separated strands from coming back together, single-stranded binding proteins go around. When the DNA is being unwound, the enzyme topoisomerase releases stress by cutting, unwinding, and reconnecting strands. The DNA splits into 2 strands, the leading and the lagging strand. The top strand is the leading strand because it goes from 3' to 5', while the lagging strand goes from 5' to 3'. The leading strand then starts to be replicated by a DNA polymerase. But before the polymerase can begin, an RNA primer made by RNA primase needs to be placed so that the DNA polymerase knows where to begin. RNA primer knows where and what to put because of transcription and translation. When the DNA is being split, RNA polymerase reads and writes it in messenger RNA. So a sequence AC, GT, GC, the A is replaced by a U. So the messenger RNA sequence would look like UG, CA, CG. It has different codons that encode for specific amino acids. Initiation begins when a small ribosomal subunit moves from the cap to the initiation site. Then transfer RNA that contains anticodons complementary to the codons on the messenger RNA. It typically starts with AUG, so the transfer RNA anticodons are UAG. At the end of transfer RNA is the amino acid that corresponds to the codon. Amino acid from the first attaches to the second amino acid, and the ribosome moves down the messenger RNA, and the next transfer RNA ends and the process continues. This is called elongation.
When a stop codon is encountered, a release factor enters the A site, and translation is terminated. When termination happens, the protein is released, then the protein is folded to have a function. Once the RNA primer has been laid, the DNA polymerase III proofreads and replicates the rest of the DNA on that strand. It starts the daughter strand of DNA on the leading strand at the 3' end because DNA polymerase adds DNA 5' to 3'. A sliding clamp protein allows DNA to synthesize long strands without falling off DNA template. To replicate the lagging strand, it is different because the strand is running the opposite way, so it is uncontinuous replication. The RNA primer is put down in different segments, then the DNA polymerase goes through and replicates in the spaces going from the 3' end to the 5' end. The segments are called Okazaki fragments. Once both strands are replicated, DNA polymerase I removes the RNA primer and fills in nucleotides that are needed. DNA ligase then seals up the fragments to make the double strand. Now there are 2 semi-complementary strands of DNA, and the cell can go on dividing like normal to create the new undamaged cell.